Okay, great. Hi, everyone. Thank you for joining us this morning. First of all, I'd like to thank all the organizers for inviting me to speak at CAMDA today. My name is Krista Ryon. I'm a researcher at the Department of Physiology and Biophysics at Weill Cornell Medicine in New York City at the Christopher Mason Lab. Currently, I am the executive director of an international consortium known as MetaSUB, and that will be the main topic of my conversation today. I've been part of the consortium for a couple years now, and I've been executive director for almost a year. So today I'll be starting off with what really MetaSUB is as a study project and consortium, and then how it's spanned from a city scale project to a global scale project in these last couple of years. All right. So MetaSUB's kind of been touched on before by Lynn, and it probably will be again throughout CAMDA. But I guess this all really started off with a question. What is around us? I'm sure some of you guys took a car or bus today to get to the conference. Some of you may have taken a train, ridden on an airplane to get here. But what is really around us when we're touching all the services around us, a subway pole, what is really there, and what are the microbes that we're interacting with? So this was really the leading question at the start of the study, examining not just our human microbiome, but the microbiome that's really all around us. So as many of you know, microbes are omnipresent. They can, excuse me, they can survive in diverse environments, from humans to urban built environments, aka cities. And we found it's important to understand the environment since, sorry, excuse me. So we found it was important to understand the environment since approximately 55% of the world population, or 4.2 billion people, reside in these cities. It is a trend that is looking to increasingly go up to almost 70% in 2050. So we're really looking that more and more people are really moving to these urban environments. And it's probably a trend that will continue on moving forward. Looking closer, we found that mass transit systems generally host large volumes of people and facilitate this constant stream of human to human and human to built environment microbial transmission, as we're always going through these high interaction places and leaving and coming back in our day-to-day lives. So with all this in mind, a project was launched in 2013, led by Dr. Christopher Mason and Dr. Evan Ashton, where they started a study to characterize the microbiome and metagenome of the built environment in New York City. So they found that New York City was this ideal place to begin to understand the urban environment, due to New York City being one of the highest population densities in major cities in the United States, therefore being a really great representation of what the urban environment really is. So Chris went out with a bunch of students, as did Evan, and they went out and swabbed 3 samples from each of the 466 subway stations in New York City, which took a whole summer, and that was a total of 1,457 swab samples. And these samples were processed using metagenomic shotgun DNA sequencing, creating one of the largest sample sets of city-scale metagenomics. Results from the study were published in Cell Systems in 2015, producing this first map of microbes at its scale. It was found that the majority of the 637 known bacterial, viral, fungal, and animal species detected were non-pathogenic and represented normal bacteria present in the human skin and human body. However, it's also found that almost half the DNA present in these subway surfaces matched no known organism at the time of the study. So because of this, there was a lot of questions to be asked. Why was there such a great deal of unknown organisms in the area, as well as the fact that the majority of the organisms that we were picking up were bacteria. So results from the study really led to the launch of MetaSUB, moving back to that, where other cities were interested in what type of microbes and organisms were inhabiting their cities. So this international effort really grew that year and led to sampling over 40 other cities that year. It also came together as an international consortium made up of experts across the fields and every continent. So the MetaSUB International Consortium was really born out of PathoMap and all of this work in New York City into this now international effort. So really diving into kind of what MetaSUB is now, going from New York City now to this international effort, it was kind of a jump. So going from sampling only one city to organizing an effort on a global scale really required the consortium to start developing standards, as Lynn said, and trying to really hone in on aims on how we're going to do this and effectively create this geospatial metagenomic map on a planetary scale. So this was started mainly to create this geospatial metagenomic map, identify antimicrobial resistant markers by synthetic gene clusters. And this study really evolved into this organization and consortium that's all really working together toward the same goal. And a lot of the components that I just touched upon, and I will as I go forth with the presentation, is this global sample collection that's been ongoing since 2016, and then developing standards for sampling as well as computational methods. So as I talked about our global sample collection, it started in 2016, and since then we've collected over 40,000 samples from cities around the globe. A major project in our consortium that takes place annually is known as Global City Sampling Day, and as you can see here, you can see a spike for each year where we really have a lot of cities just kind of come together and collect a mass amount of samples on this one day. And we have representation from a lot of cities in Europe, Japan, Asia, North America and South America, and some cities in Africa. And since then, it's been expanding, and as of now, we've had over 180 cities participate in our global since 2016, and we hope to continue to increase that number and really reach into all the global cities in the world in order to really determine what microbes are existing in each urban environment and how they differ and how they're similar, and how they translate throughout the years. So as I said about Global City Sampling Day, in 2016, it marked the first annual event of this simultaneous sampling day, where scientists, researchers, and even trained citizen scientists collected samples from mass transit systems around the globe. So this sampling effort is really what largely makes up the MetaSUB data set that we kind of all know of. And that data set has been generated from years and years of sampling effort. But since then, MetaSUB has really expanded into several different projects across many different fields. And sampling has not just been done in urban environments and subways, but of monuments, sewers, beaches, airs, and even medical environments. So since 2016, we've really tried to expand MetaSUB into not really being MetaSUB as in subways, but kind of figuring out what type of microbes are really inhabiting different spaces. And as Lynn touched upon in her presentation, and I did a little bit earlier, creating the standardization as we progress with sample collection became increasingly important, making sure that since we cannot potentially standardize how a lot of the variables within each city, and even time and day or temperature and things like that, that standardizing actual sample collection, as well as processing through automation and sequencing and batch in order to create less variability within the processing of our sample set. And then we try to create more standardization into our downstream pipelines. So as I mentioned, we have moved from subways which we still very much sample, to monuments and sewers and other sort of projects. We have several ongoing projects in MetaSUB, and I'll touch upon 2 today, just very briefly. So these are our MetaSUB and MetaCove projects. So our MetaSUB study was launched in 2016, and it focused on the periodic analysis of sewage and wastewater systems in order to detect high-risk pathogens, as well as identify antibiotic-resistant pathogens in wastewater. The study was initially led by Dr. Krista Ryon in the Institute Pasteur. But more recently, in 2020, we've started a study through the NIH Rapid Acceleration of Diagnostics, or RADx initiative, where we're taking weekly wastewater samples with Miami-Dade County in order to test for SARS-CoV-2 and its variants. This study is ongoing, and we are using currently RT-qPCR, targeted amplifying approach, with the NEB Arctic workflow and shotgun metatranscriptomics for a more comprehensive look for viral RNA. And through this study, we aim to really look at more of a surveillance approach with these emerging pathogens and microbes that are in our wastewater. So as we can see here, there's a very close correlation between sewage and population metrics, as in copies of SARS-CoV-2 found in positive cases of coronavirus. And then we hope moving forward to use the wastewater to detect variants of concern and track other respiratory viruses. Excuse me. So moving on from wastewater and kind of backwards toward the beginning of the pandemic. So for MetaSUB, we were on this track of really sampling subways and looking at metagenomics. But with the emergence of COVID-19 pandemic in 2020, we came together and we started sampling services during the peak of COVID-19, during the lockdown, and specifically city lockdowns and subway stations. So as a lot of these cities, as we all know, were locked down, a lot of subway stations shut down, a lot of transit systems shut down entirely or became not as frequent. So we went and we were starting to swab high touch surfaces like ATMs and park benches. And in this specific study, we've started to collect, we collected almost 4,000 samples in 27 cities in order to kind of track the emergence of COVID-19 pandemic throughout 2020. So this processing is ongoing, but we do have a pilot that we did in New York City where we did collect 86 New York City subway samples from handrails, kiosks, and the floors of Grand Central and Times Square early on in the COVID-19 pandemic. And we found the results indicated that high transit surfaces are not likely to harbor significant levels in the early phases of the pandemic in New York City. And this is through SARS-CoV-2 reads. But we're continuing analyses on this, and we hope to publish some more results soon on the entire data set. So moving back to more present time, last year, after a bunch of sampling and analyses, we published our first, we had our first major publication, it's still highlighting samples collected from 2015 through 2017 in 60 cities, amassing to over 4,000 samples collected. So this study highlighted kind of a few things for the consortium, but mainly this core microbiome that we found distinctly within all the cities that we did sample. So to our knowledge, the study represents the first extensive global metagenomic study of urban microbiomes. The study reveals a consistent core of urban microbiome across all cities, annotated according to gram stain, ability to form biofilms, and whether bacteria is a human commensal species. So, and we found that most of the bacteria in this core was associated with the human skin or respiratory tract, one of which is Cutibacterium acnes, a gram-positive bacterium closely linked to human skin. So as we're, outdoors, shedding skin, shedding respiratory, droplets. This is expected to be quite common. We also looked at the distribution of microbial species across the global urban environment. Specifically, we looked at the single type of microbial, excuse me, we looked at the biodiversity across the cities and species prevalence, excuse me. And we used this to determine separate sets of taxa based on inflection points of the distribution as a subcore and true core species, as we saw on the previous slide of the core. Okay. All right, and just to keep with time, I'd like to touch briefly upon how we're looking also at the possible link between the environment and human antibiotic resistance, leading to transfer of antibiotic resistance and genetic determinants from clinically relevant microbes to human commensal microbes. And we also looked at the abundance of these AMR genes in our data set. So for AMRs, the goal for these analyses was to try to predict antibiotic resistance from gene sequences, mapping reads to known resistance genes using meta-res ontology and quantifying their relative abundances. So over 2,000 samples had some sequences aligned to the AMR genes, but no consistent core set of AMR genes was identified in these data. So despite these low abundances of genes themselves, some samples contain sequences from hundreds of distinct AMR genes, and clusters of high AMR diversity were not evenly distributed across cities. As we can see here, some cities had more resistant genes identified than others, such as Bogota, where other cities had bimodal distributions, San Francisco, and some were very, very few. So also using these data, we were able to create an online map where you can see these different microbes and characterizations of different cities. So I invite all of you to go here to metasub.org/map to view that and all this data. And special thanks to Dr. Jen Maiden and Dr. David Danko for putting this together. And in brief, just this paper and the study really represents the first comprehensive survey of the built environment. And we really aim to continue this endeavor of characterizing the microbiome of not just our cities, but our current environment through air, sewage, etc. So especially in this day and age where we're not just looking at pathogens, but commensal species, and that could perhaps give us insight into how we can coexist with our everyday microbes and identify potential emerging pathogens. Thank you. Thank you to everybody at the MetaSUB Consortium, Mason Lab, and CAMDA Organizing Committee. Thanks. Are there any questions? I'll stop sharing.
You're good. I mean, I can. Can you hear us?
I can hear you a little bit.
Okay. So if you go to the Q&A session on the right-hand side of your screen, there's a tab like chat and Q&A. So if you click into that, there should be people asking questions there.
Okay. Should I read the question?
That'll be great. Yes.
The MetaSUB effort sounds fantastic, but I'm curious if the effort is mainly exploratory, if there are specific insights this effort is hoping to uncover, or is mainly the collective data and let subsequent studies extract insights such as uncovering AMR. I'll say that we definitely have specific aims through creating this geospatial metagenomic map and seeing what microbes are really inhabiting our urban spaces in terms of surveillance, in terms of tracking AMRs. But some of it is exploratory in the sense that a lot of this stuff is novel, and we really aim to kind of capture a wide breadth of like what type of microbes are in the area. and then kind of going further in there. So for our COVID-19 study, we are doing mostly metatranscriptomic sequencing in order to kind of look at the gene expression as well as track for other viral pathogens that we could possibly pick up. And let me know if that answers the question. I know that was kind of